$50,000 a year for the drug alone. Yes, ma'am.
I was going to ask about the Tysabri. Have you guys we read a forum online about how they think that there might be a link to getting cancer after like the 3 or 4 years of being on it because of it lowering your immune system.
Okay. So the question is that online there was was it a blog or something?
It was an MS forum. I don't know.
Okay. So it was an MS forum that the issue was brought up was whether long-term use of Tysabri, 3 to 4 years or longer, might be associated with an increased risk of cancer based on the fact that it may suppress the immune system. It actually does not suppress the immune system. That's something that many people believe, but in fact, general infections outside of the brain are not increased in patients on Tysabri. So if you compare patients on Tysabri to non-MS healthy controls or to patients in the study, we don't really see a difference in routine infections. What we do see is after the infusion, there's patient, about maybe 10%, 20% of patients report fatigue, about 8 to 10% report headaches lasting for a day or so. And there does appear to be a signal suggesting there might be an increased risk of shingles in the population when we look at the TOUCH data, which is the post-marketing surveillance safety data set. But in terms of cancer, we've not seen any signal there that there's something unusual going on. We have seen cancers in patients, but when we look at the 80,000 patients worldwide and we match them up against the Medicare database, where we can match up against age-matched controls, it's the same number as you would expect. And so at this point, I would say it's unknown. I would also say that I've seen one cancer here. I know of 3 other cancers, serious cancers, lymphomas primarily, which are the ones we're most worried about. There's been 4 reported total so far. 2 occurred, so there's 5 reported. 3 occurred within a month or 2 of starting the drug, which doesn't fit the biology. That's not how lymphoma works. So those are probably coincidental. 2 occurred when the patient's been on it for 6 months or longer. So those are more worrisome. But the prevalence of lymphoma in the population is about 1 in 40,000. And there's 80,000 patients on the drug, so that's about what you would expect. So I would say that it's an open question. But the advantage of that drug, from my perspective as a physician, is that it's the only drug that has 100% safety monitoring mandated by the FDA. I would like to see that for all new drugs coming in the marketplace. I wish they would have done it for Gilenya, that we would learn faster. We'd know, we'd be able to give you better data. Unfortunately, because that was not mandated for Gilenya, Novartis, the parent company, has not really implemented a proactive safety monitoring program, so they rely on random reports from physicians. And we know from experience that they only pick up a small percentage of the actual serious adverse events that are occurring. So it takes us much longer to recognize there's a problem. So this is something I would urge you to advocate for Congress to do something useful, require that we actually put in systems to monitor what these new drugs do to people over time and look at 100% ascertainment. Yes?
Is there any evidence stating that Tysabri can interfere with hormonal changes in the body?
So the question is there any evidence that Tysabri can interfere with hormonal changes? No, not at this time. Tysabri is a remarkably selective drug. It only binds with one protein in the body. It's like rituximab, only binds to one protein in the body. And in both of those cases, that protein is only expressed on immune cells, with one exception, which is sperm, in the case of Tysabri. But no, the Tysabri binds to a protein called alpha-4 integrin, and that's a protein that's critical for lymphocytes to migrate into the brain. Lymphocytes are the key immune cell that drives the disease. And it does that so well that the brain becomes an immunocompromised organ, and that's why we see PML. So prior to Tysabri and alemtuzumab and other drugs, PML was a disease we only really saw in cancer patients and in AIDS patients. But now we have a model where the immune system in the rest of the body is essentially normal, but in the brain it's deficient, and so that allows this opportunistic infection to occur. But we've not seen any evidence that there's any other hormonal changes with it in the studies. And what data I'm using there is just the data from the clinical trials where we actually had a comparison group where people were randomized. And there wasn't any signal that there was anything else going on. Okay?
How about rituximab? How does that work?
Okay. Rituximab is another monoclonal antibody like Tysabri is. So you understand when I say monoclonal antibody what that is? Okay. So antibodies are produced by B lymphocytes. So you have several different kinds of lymphocytes in your blood, and one of them is called a B lymphocyte. And the B lymphocytes are particularly prominent in the spleen and lymph nodes. So when you feel a lymph node, that's mostly B cells you're feeling. And they're cells that have a receptor on their surface called immunoglobulin. And when they're activated and become mature, in other words, they're stimulated so they find a target they can bind to, then they proliferate and they begin to actually secrete that receptor as antibody that you find in your blood. You produce billions of different kinds of antibodies, and each one of them are very, very targeted to a very specific three-dimensional structure. And as a result, they're like a lock and a key, but even more refined than what we currently have. Humans can't make systems that are quite as selective as these molecules are. They are remarkably selective in how they work. So they become sort of a guided missile for that particular structure, whatever they're aimed for. We can now in science, and I used to do this in the laboratory, which is kind of tedious, is you can immunize a mouse with a protein that you want to target in a human. And the mouse will reject that protein by making antibodies to it that bind to it and then causes it to be degraded and removed from the system. We can then take the mouse immune system and isolate it from it, the cell, the single cell that makes the antibody that binds to the protein that we're interested in. We can grow that cell into billions of them and have it produce the antibody in pure form. And that's what these things are. These are antibodies that were generated to a specific target that are pure. There are only one kind of antibody in them. That's why we call them monoclonals. When you do an electrographic separation, you get one peak. And rituximab is one of those types of molecules, and it's targeting a protein called CD20. And CD20 is expressed on B lymphocytes only. It's not expressed anywhere else in the body, only on B lymphocytes. And it's expressed on B lymphocytes when they're immature to the time they become memory B cells. And then memory B cells can actually mature one more step where they become plasma cells. And plasma cells are these factories that just crank out tremendous amounts of antibody. But this drug does actually not kill plasma cells. So it doesn't lower your antibody levels. Your antibody levels, so if you've been immunized against influenza and you get treated with rituximab, you still have antibodies to influenza. And your plasma cells will continue to produce that because they're long-lived, decades, maybe longer. But it does remove the B cell. And so this drug was actually a very interesting experiment because at the time that it was tried in humans, we all predicted it would fail because we said MS is a T-cell driven disease. And if you've read on the internet, you know, that's sort of the blog that it's a T-cell driven disease. So this was a test of hypothesis that where you would not remove the antibodies, so you wouldn't remove antibodies being part of the process, you just remove the B cell. And it turns out it's one of the most highly effective therapies that we've ever seen. It shuts down the disease between a 60 and 80% reduction of relapse rate, which is as good as any other drug we've seen. And because it only removes B cells, it leaves all the rest of your immune system intact. So the part that helps you resist bacterial infections and most viral infections and cancer are left intact. Those systems are not damaged by this process. Just your ability to make new B cell or new antibodies. And it appears that in human disease, the autoantibodies that participate in human disease are often produced by B cells. So this drug is also used in rheumatoid arthritis and lupus and other inflammatory conditions. So it actually fundamentally changed our understanding of human disease. It turns out you're not like mice. Mice, it is a T-cell driven disease, and we can prove that because we can purify the T cells and put them into another mouse, and that mouse will get the MS-like disease. We tried to get volunteers to do that in humans, but we couldn't get very many people to volunteer for that. So this is an indirect way of getting the same question. So we actually have done clinical trials with monoclonal antibodies that targeted the T cells. They didn't work. They failed, both anti-CD3 and anti-CD4. And most immunologists don't know about those trials because they're published in clinical journals. Immunologists don't read clinical journals. And so there still is often written that this is a T-cell driven disease. But in human biology, all of these therapies, all of them, they may affect T cells, but they definitely affect B cells. So the one common denominator under all the therapies so far is B cells. And I would argue with Copaxone, some of your colleagues disagree with me, but we've published papers where we can treat animals with Copaxone and then purify their B cells and put them into another mouse and show that those B cells will protect that mouse just as though they got the Copaxone directly. And that's pure cultures sorted by flow cytometry. And we published that, but I don't think anybody in your company ever read it. And the next slide.
They were
Yeah. They don't read English. So let me just come back to these two a little bit. These are both pills. They are drugs that we still don't understand how they work, and in fact, they were developed empirically. So empirically means that one of my colleagues thought it might be a good idea to try one of these drugs and just put it into mice, and it worked. And then they went on to humans. Now, the case with laquinimod is slightly different. Laquinimod is a drug that's been modified from another drug called linamide. And linamide was an older drug that was available in pharmacies for other purposes, and was tried in MS, had initial benefit, and then we did a large phase 3 trial, and we found that it could induce myocardial infarctions and serositis, so an inflammation of the lining of the lung and things. So we had to stop the study as a result. And so a company in Sweden took that molecule and then began to modify it, make substitutions to try to find something that would still benefit MS but wouldn't cause those side effects. And I didn't think they could do it, but it turns out that they did. So they made this drug. We've not seen any of those side effects with the drug, despite almost 5,000 patients now being treated with the drug. And it has a remarkably good safety profile. And as I mentioned at the American Academy of Neurology meeting in Hawaii about a month ago, the data was presented, and this had a significant effect. It met its primary outcome measures and showed a very nice effect on brain volume preservation, which for me is a very important issue. And it did that without any suppression of the immune system and any increase in infections. So these drugs, as I said, are completing their phase 3 trials this year. And this one should be done maybe mid-July. And then it'll go to the FDA. Presumably, I hope the company moves rapidly to get to the FDA. And these both are on fast tracks. Theoretically, they could get approved in 6 months. The FDA has never approved a drug in 6 months that I know of. So it's not likely. But nevertheless, by next year, I think they could be out. They will. Yes, ma'am.
What companies are sponsoring those?
BG12, I think Biogen bought the rights to that, didn't it?
Biogen.
Yeah, but it was developed by a company in Sweden that I don't remember the name of. I consulted for them, but I don't remember the name of them. So I've actually consulted for all the companies that make all of these drugs, so that's a conflict of interest. So I'm promoting them all equally. Linamode is actually now owned by Teva Pharmaceuticals. And again, was bought from I'm sorry, I got it right. BG12 is actually developed in Germany. I don't remember the name of the small company. It's now bought by Biogen. Liquinimod was developed by a Swedish company. It was now bought by Teva. And that actually is necessary because, again, as I said, the cost of taking it from first-in-man trial to FDA approval is about $1 billion. Small companies can't afford that, let alone the NIH or us. So, any other questions about these?
How do those compare to the rituximab in your estimation?
Well, these drugs, look, the question is how do these two drugs compare to rituximab? Rituximab had a 60 to 80% reduction in relapse rate, depending on which study you look at, whether you look at ocrelizumab or rituximab. This drug is 27%. This drug is 58%. I don't have any other, and that's based on a newspaper report, so I don't have the scientific data. I don't know any of the other outcome measures for this particular drug. Liquinimod had a large effect, basically as large as we've seen with Tysabri and rituximab, on brain volume preservation and on disability. So it had about a 30% reduction in disability risk rate, which is surprising to us. We thought that relapse rate would go with that, and these are the first drugs to really separate relapse rate and disability.